Okay, so now that we've talked about the general idea of genetic engineering, we're going to talk about how you are going to use this in your lab next week. This is on page 50 of your notes, and so you should take the notes in your lecture notes. You will have a pre-lab quiz on Monday, and it will be open notes, so you will be allowed to use any notes you take today in class on that quiz. Okay, so here we go. The overall goal of your lab experiment for next week is going to be the following. You are going to use genetic engineering techniques to insert a specific gene, in this case the GFP gene, into E. coli. So there you can see in the diagram, right, here's our plasmid DNA that contains the gene we're interested in. In this case, the plasmid is called pGLO, and we'll talk about it in a little bit. And we are trying to get it into the E. coli so that they can then transcribe and translate this gene and make lots of protein that we want to produce, in this case the GFP protein. So let me give you a little background first. GFP stands for green fluorescent protein. You can actually see some pictures of it occurring naturally in organisms in this slide. GFP is naturally produced by a jellyfish. It's actually called Aquaria victoria. It was first discovered in the 1960s, and it's a source of bioluminescence. When you shine UV light on the jellyfish, they actually glow green. Just for interest, this is the structure of the GFP protein. You kind of see some of the aspects of things we've talked about in terms of protein structure in the picture, but really it's just for interest. You don't need to know anything about this picture. I just threw it in there so you would know what the protein itself looked like. Kind of has like a barrel shape. Let's talk about, first of all, why GFP or bioluminescence in general would be useful in nature, and from there we'll talk about the lab. So in nature, bioluminescence can be useful to organisms for a number of reasons. It could help them to attract mates. For example, I believe fireflies, when they're trying to attract mates, they will shine their lights in sort of a species-specific pattern to kind of attract the opposite gender. It might simply be for visibility, to see their food. And as you can see down here in the picture, it can also be for defense, because if an animal is able to spray some sort of bioluminescent substance towards a predator, it gives them the time to then escape. What scientists have now found, though, is that if they can isolate the GFP protein, they can really use it in a lot of useful ways in scientific research. So we're going to look at some of that. And you can write these in at the bottom of page 50. Because GFP is visible, so easily visible, it is a way to tag the cells. So potentially what I can do is, if I can have a cell express or produce GFP, then what I'm going to be able to do is, and I want to know if I want to find a particular cell, then it's a way for me to actually visually see that cell. And that's what you see in the upper right corner here, is cells that have been tagged to produce GFP. That way I know those particular cells are present if I'm looking for them in an experiment. GFP can also be used as what we call a reporter gene. What that means is, if I can attach the gene for GFP to another gene, then I'm also going to be able to see when that other gene is being expressed, because if they're all tied together, then if I'm making GFP, then I must have also transcribed the other gene. And so that's what we mean when we say it's a reporter gene, that is linked to another gene to show if that other gene is expressed, because it might be harder to detect than GFP. Lastly, GFP has actually been expressed in entire animals, and you see a variety of pictures of that at the bottom of this slide. On the very, very right, you see transgenic mice. So those are mice that have actually been genetically engineered to produce GFP, and scientists are using them to study disease. There are some others that have been used, made more for, I guess you could say, commercial purposes. In the middle, you see glowfish. Those are fish that actually express GFP. Technically, you can buy them commercially, but they are not available in California. They actually won't ship to California. And then lastly, on the very left, you have Alba. Alba is the green glowing bunny. She was actually a bunny that was genetically engineered to express GFP in all of her cells, and so she did, in fact, glow green. If my information is correct, she originally was some sort of partnership between a lab and a French artist, but at the last, and it was supposed to be part of an art display, but at the last minute, the lab really felt kind of uncomfortable about having, done this, and so I think Alba was not actually put in as part of the art installation and such. But she did, in fact, exist, and is an interesting, example of how GFP has been used. What they've done too now is they've actually been able to take it beyond just GFP. And they've been able to mutate the gene for GFP to create other colors. So now there is YFP and RFP and BFP, so yellow fluorescent protein, blue fluorescent protein, red fluorescent protein, and they are all mutations of the GFP gene. GFP also has another piece of notoriety, I guess you could say, or fame to it. Not too long ago, in 2008, 3 scientists were actually awarded the Nobel Prize in Chemistry for their work on GFP. And at the end of this PowerPoint, you will watch a video about that, just to kind of give you some context of sort of the evolution of the discovery of GFP and what these particular scientists really did and why it was so groundbreaking. All right, so let's talk about our lab. In your lab, you're going to use the pGLO plasmid. Technically, if a scientist creates a recombinant plasmid like pGLO, you can name it whatever you want. So in this case, they named it pGLO. And the plasmid in this case has 3 genes on it that we're interested in. So I'm going to walk through those, and I'll talk you through this diagram here. First, of course, is the gene we're interested in, which is the GFP gene. It codes for the GFP protein. So ultimately, that's what we want to get the bacteria to make. Secondly, we have the BLA gene. The BLA gene codes for an enzyme called beta-lactamase. And what beta-lactamase does is it actually destroys the antibiotic ampicillin. Normally, antibiotics are supposed to kill bacteria. So if a bacteria has this plasmid, they actually have essentially an antidote to the poison. They have a way of protecting themselves against the specific antibiotic ampicillin. Lastly, we have the ARAC gene, and the ARAC gene codes for the ARAC protein, which I'll explain its significance a little bit later. The last thing you might be wondering about in this diagram is this thing called ORI. This is called the origin of replication. You don't really need to worry about what that is right now, so just don't worry about it. Okay, so moving on then, let's talk about how is it going to work. We've already talked about how we're going to get the plasmid into the E. coli. Let's talk about then how does it work. How does this plasmid work? How are we going to express the GFP gene in our E. coli? To give you a little background, I have to tell you about what's naturally occurring first, and then we'll talk about how scientists were able to change it. We talked about operons already in bacteria, and one example of an operon is the Arabinose operon in bacteria, which normally looks like this. You have structural genes, the ARA-A, ARA-B, ARA-D genes, and they code for enzymes that are going to digest the carbohydrate Arabinose. Now, usually what happens is the protein, ARA-C, is actually bound to the operator region and prevents transcription. So take a minute and talk to your neighbor then. Would this be an inducible operon, or would it be a repressible operon? Because the ARA-C is normally bound to the operator, it's normally off. So this would make it an inducible operon, meaning we have to switch it on when we need to. So usually the Ara-C protein binds to the Arabinose operon operator region and therefore prevents transcription. But when I have the sugar Arabinose around and I want to digest it, now I need to essentially switch on this operon. I need to unlock it, right? So that's where the protein Arabinose comes into play. When Arabinose now binds to the Ara-C protein, it's going to change its shape. Now the Ara-C protein can't bind to the operator anymore, and that allows RNA polymerase to hop on, transcribe these genes, and therefore make the Ara-A, Ara-B, Ara-D enzymes, which will then digest Arabinose. Okay, all of this is only important because it gives you a context of what was naturally occurring in the bacteria. What scientists did for the pGLO plasmid was they took this operon and they adjusted it. And here's what they did. So if you look on the following page there, you have a diagram that says expression of green fluorescent proteins. We're going to look at that diagram now on the top of page 52. So this is a modified Arabinose operon because the idea was they said, well, let me use the way that this operon is controlled, but let me now put in the gene I want to be expressed, not the Ara-A, Ara-B, Ara-D. So scientists modified the Arabinose operon in the pGLO plasmid so that now it would express the GFP gene instead. Same control mechanism, right? Normally Ara-C now binds to my operator, prevents transcription. But what's after that is the GFP gene. So now, when I add Arabinose, what's going to happen is that Arabinose binds to Ara-C. Ara-C can no longer bind to my operator. Now, what I transcribe is the GFP gene, and I'm translating that too. So in the end, if I want this bacteria to be able to express GFP and glow, I have to add Arabinose. Okay, so let's move on then to talk about some more specifics about your lab. Remember we talked about the fact that transformation's not 100% successful. Inevitably, when you do a transformation reaction, you're going to have some cells that got the plasmid, in this case pGLO, and some that didn't. So selection is the process where we figure out who got the plasmid and who didn't. And to do that, we're actually going to use what we call a selectable marker. A selectable marker is basically a trait that's going to help me identify which cells got the plasmid and which ones did not. Now, in our experiment, we're going to use that BLA gene as our selectable marker. Think about why for a second. What does the BLA gene code for? So take a minute, talk to your neighbor about why. Why would the BLA gene, the presence of the BLA gene, allow us to figure out who got the plasmid and who didn't? Let's just review to kind of give you something to go on. What does the BLA gene code for? It codes for the beta-lactamase protein, right? And what does that protein do? Remember, it destroys the antibiotic ampicillin. So now the big question, how does it serve as our selectable marker? Well, an E. coli that doesn't have the pGLO plasmid won't have the BLA gene, which means it doesn't have the ability to digest and destroy ampicillin. So if you were to put this bacteria in the presence of ampicillin, it would die. However, if you have a plasmid that has the pGLO plasmid, therefore has the BLA gene, now it has beta-lactamase. So now it's not going to die when you put it in the presence of ampicillin because it has a way to protect itself. That's how this gene is going to be our selectable marker, because only cells that have the pGLO plasmid will make beta-lactamase and then are therefore resistant to ampicillin. They will live in the presence of ampicillin, while cells that do not have the pGLO plasmid are going to die. So basically what they do is they give all the cells the poison and they see who lives and who dies. The ones that live are the ones that got the plasmid. All right, so let's also now talk a little bit about, well, how are we going to grow the E. coli? Because once you've done this and you give them the poison, you basically want to see who's going to be able to grow in that scenario, right? So a little bit of background for you. Like anything else, bacteria have specific nutrient requirements. So we grow bacteria on a type of food called LB agar. It actually stands for Luria bertani agar, but you don't need to know what it stands for. Just know it's called LB agar. LB agar contains all the nutrients, the amino acids that the E. coli need to survive. So it's food for the bacteria. If I put bacteria on a plate of LB agar, they will grow very happily. It's like a buffet for them. What I can do though is I can also add other stuff to my LB agar, like antibiotics. So in this case, if I wanted to see if E. coli could grow in the presence of ampicillin, I could add LB agar plus ampicillin. So it's like food plus poison. And then it becomes selective, right? Because now only some E. coli are going to be able to grow on the LB agar, the ones that have the ampicillin resistance, whereas the other ones would die. And that's basically what you're going to be doing in order to figure out who got the plasmid and who didn't. So we have this diagram right here. You can see in your lab what you're going to do is go through the whole transformation process. You're going to add the calcium chloride, you're going to do the heat shock. So you're going to add the plasmid, obviously, and hope that some of the cells got the plasmid. So at this stage, what you've got is you've got E. coli cells that have gone through the transformation process, and some of them got the plasmid, some of them didn't. We got to figure out who got it, who didn't. So we can do one of two things. We're going to take some of those E. coli cells and we're going to grow them on plain LB agar, right? It's just food. So now what you need to ask yourself is, and talk to your neighbor about it for a moment, is who grows on that plain LB agar? The transformed ones, the untransformed ones, or both? What we can also do is take some of those cells, and now we're going to put them on LB agar with ampicillin. Again, now who grows? Transformed, untransformed, both, neither? That's the question, right? Well, remember, LB agar is just food. So if I put my E. coli cells that have gone through the transformation process on plain LB agar, everybody grows, right? Transformed, untransformed, it doesn't matter because it's just a buffet. So all E. coli will grow. The difference is the second plate. If I put them on the LB agar with ampicillin, now only my transformed cells grow, right? Only the ones that got the plasmid are going to grow. The untransformed ones can't survive, and so they die. That's how you're going to do the selection process, because you're not going to know who got the plasmid and who didn't. So you're going to put them on these plates, and by seeing who grows, then you know who got the plasmid. All right, with any experiment, we have to have controls, right? We need to have things that sort of make sure that we've accounted for certain factors. So you're going to have 3 controls in this lab. The first one is you're going to take E. coli that we did not give the plasmid to, so we called it the minus DNA sample, and we're going to grow it on plain LB agar. Should it grow? Yeah, it should, right? It's just food. So why do we do this? We want to make sure there's nothing wrong with our E. coli, right? We want to make sure they're capable of growing, because what if we had some sort of problem E. coli that couldn't grow anyways, and then we thought that we just hadn't done the transformation correctly, when in fact they just couldn't grow at all. So that's our first control. Our second control is, again, we're going to take E. coli without the plasmid, so we call that minus DNA, and now we're going to put them on LB with ampicillin. That's abbreviated LB-amp. Should they grow? No. In this case, they should not grow, right? Because they have no resistance. They have no protection against ampicillin. They should die. So then again, why do we do this? Well, we're assuming that before we did the transformation, they were not resistant to ampicillin, that by doing the transformation, we gave them that quality. What if that's not true? What if your E. coli are already somehow resistant to ampicillin? So then you think you've done something, really haven't, right? We need to make sure. So this allows us to make sure that the E. coli we started with that did not get any plasmid weren't already resistant to ampicillin all by themselves. Lastly, we are going to have our third control, which is grow the E. coli with the plasmid on LB with ampicillin. Now we're thinking, should they grow or should they not? Yeah, they should, right? If they got the plasmid, they should grow. Will they glow? No, why not? Because we didn't give them Arabinose. Okay, so why is this a control? This is a control because these cells will have gone through the whole transformation process with the plasmid. We want to make sure the transformation didn't kill the E. coli. Because if it did that, that would also be really bad. Right, so ultimately our experimental plate is the one where we grow E. coli with plasmid on a plate with LB, ampicillin, and Arabinose. So what you're going to do is you're going to do the transformation procedure. You're going to have these different plates, plus your last plate, your fourth plate, which is your experimental plate, which is E. coli with plasmid on LB with ampicillin and Arabinose. And you're going to see what you get the following day. Because we have to give the E. coli time to reproduce, to show us who got it, who didn't. The last thing we're going to talk about then is a quantitative measure of how well this whole experiment worked. And that's the idea of transformation efficiency. There isn't really a specific spot for you to write this in your notes. I would say just write it in at the bottom of page 52. Transformation efficiency is basically, it's a way for us to numerically measure how well did our transformation work. Ideally, we want it to have worked really well. And so we'll get the details a little bit later after you've done your lab, but effectively what a transformation efficiency measures is how many bacteria got transformed by 1 microgram of plasmid DNA. So the general calculation we're going to use is we're going to say, okay, how many cells are growing, we assume, on our selective plate, right, compared to how much DNA we originally put on that plate. Ideally, what we want to see is more cells growing for the amount of DNA spread on the plate. That means your transformation efficiency would be really high. And so when scientists do experiment, that's one of the things that they sort of try to adjust to try to get the best transformation efficiency. And after you get your data back from this lab, that is one of the calculations that you'll be doing. But we'll go into the details of how to do that a little bit later. So for now, just sort of know what transformation efficiency is, but don't worry about the math behind it. In a later lecture, we'll talk about that. Now that you've finished this lecture, then you're going to do a couple different things. Number one, you're going to watch a short video about that 2008 Nobel Prize winning trio who worked on GFP. And for your homework, which is essentially going to be due on Monday, is you need to, number one, set up your lab notebook. That's going to be the standard title, intro, paste in your procedures, make a flow chart for your procedures on the left-hand side, and that's going to be it in terms of the setup for your lab notebook. You're also going to want to study for your pre-lab quiz, which will again be open note, also on Monday. And all of that will get you ready for the lab, which will happen on the next block day. Okay, so go ahead and take some time to do that.